Good morning, dear students. Today's topic of discussion is fertilization. So, we will study the fertilization under headings of definition, location, and stages of fertilization. So, fertilization can be defined as the process of fusion of two mature gametes. From the female side, we have the mature ova, and from the male side, we have the spermatozoon. So, when these two mature gametes fuse, they result in formation of single cell stage called zygote. So in this figure you can see the male pronucleus and the female pronucleus, they are fusing and they are forming a single cell called zygote. Why this process is called antithesis of cell division? Generally we see that the precursor cell or the undifferentiated cell will divide and differentiate to form highly specialized cells. But in case of fertilization, already we have two highly specialized cells. They are fusing and they are forming an undifferentiated single cell called zygote. That's why it is called antithesis of cell division. Then the next is location. So what is the site of fertilization? Fertilization occurs in the lateral one-third of the fallopian tube in the ampullary cavity. Then we will study the mechanism of fertilization under three headings. First is approximation of gametes. In this we will study the journey of spermatozoa and ova to reach the site of fertilization. Then the next step is contact and fusion of gametes. The spermatozoa, in order to fuse with the female gamete, has to overcome three barriers, and before penetrating these three barriers to reach the oocyte, it has to undergo prior preparation in the form of capacitation and acrosomal reaction. So that will be studied under this heading. And immediately after the fertilization, the events that are occurring will be studied under the heading of effects of fertilization. The first step of fertilization is approximation of gametes. Means spermatozoa and ova has to reach the site of fertilization. So first we will see the journey of spermatozoa. Okay. So after the act of coitus, the spermatozoa are deposited in the vagina. So they have to travel from the vagina into the cervix, then to the uterine cavity, and then through the fallopian tube it will reach the site of fertilization. So during its journey in the female genital tract, there are some factors which will facilitate its transport. So what are the factors which will help the transport of sperms to the ampullary cavity in the female genital tract? First is the chemoattractants released, then the second is prostaglandins secreted from or the prostaglandins present in the semen. They will cause powerful uterine contraction. Simultaneously, at the act of coitus, oxytocin is released from the neurohypophysis of female partner. It will further aggravate the contraction of the uterine musculature, and due to such successive uterine contraction, a negative pressure is created or vacuum is created which will suck or aspirate the sperms into the uterine cavity. So these are the factors responsible for transport of sperm from vagina to the cervix, then to the uterine cavity, then to the fallopian tube, and finally it reaches the site of fertilization in the ampullary cavity. At the time of ejaculation, about 200 to 300 million sperms are emitted, and these 200 to 300 million sperms have to travel through the female genital tract. So when they are traveling, most of the sperms are obstructed by the constriction of the cervix in the form of internal os. Then the constriction is offered by the ostium of uterine tube and the cilia of the fallopian tube also act as barrier, and when these sperms are overcoming these barriers, most of the sperms will die within 24 hours of ejaculation, and only 300 to 500 sperms will reach the site of fertilization. So is it clear? At the time of ejaculation, 200 to 300 million sperms are emitted. But when they are reaching the site of fertilization, most of the sperms will die within 24 hours of ejaculation, and only 300 to 500 sperms will reach the site of the fertilization, and others will die due to the constrictions or barrier offered by internal loss of the cervix, ostium of the uterine tube, and the cilia of the fallopian tubes. Before fusing with the oocyte, the spermatozoa has to penetrate three barriers covering the oocyte, and for that it needs to get capacitated. Means there is capacitation followed by acrosomal reaction, which will enable the sperm to penetrate the three barriers covering the oocyte. So what changes occur here? Capacitation is the final step of maturation of the spermatozoa in the female genital tract, and it takes about 7 hours. When the sperms are exposed to the female genital tract, the seminal proteins, so these are the seminal proteins and few surface glycoproteins which are covering the plasma membrane over the sperm head. These are antigenic coating on the surface of sperm head, and they are involved in immunological reaction between fertilizant derived from oocyte and anti-fertilizant derived from spermatozoa, which is species specific interaction. So during capacitation, these glycoproteins are removed from the plasma membrane over the sperm head. Okay, so what is capacitation? It is the removal of seminal plasma proteins and surface glycoproteins covering the sperm head. Now it is followed by acrosomal reaction. So what is acrosomal reaction? Before that we will see the structure of sperm head. Okay, so this is the sperm head. This green color is the nucleus of the sperm, which has haploid number of chromosomes. Then this is the actin filament. It is the contractile filament present in the sperm head. Then just in front of it we have the acrosomal cap, which is bilaminar. So the inner coat of the acrosomal cap has the binding protein, and the outer layer of acrosomal cap has proteolytic enzymes such as hyaluronidase, acrosin, and acid phosphatase, and this sperm head is covered by plasma membrane. So which are the membranes present in the sperm head? First we have the nuclear membrane covering the nucleus. Then we have the bilaminar acrosome. So this acrosome is derived from Golgi apparatus, and it has inner layer which has binding protein, and it has outer layer which has proteolytic enzymes, and the outermost membrane is the plasma membrane covering the sperm head. Okay, so in all there are four membranes. So what happens in the acrosomal reaction? There are multiple contacts of the acrosomal outer layer of the acrosomal membrane with the plasma membrane. Now already during capacitation these all glycoproteins, surface glycoproteins are removed. So because of that, with the help of exocytosis, these all proteolytic enzymes are released from the acrosomal cap or outer layer of the acrosome. So these proteolytic enzymes will soften the barrier over the oocyte. Then there is polymerization of globular actin. So this actin filament, it will form a fine needle like filament at the tip of the sperm, which will help in piercing the membranes of the ovum. So these two events will occur before crossing or penetrating the barriers. Okay, which are the two events? Capacitation and acrosomal reaction. So what is capacitation? In short, it is the final step of maturation occurring in female genital tract. It takes 7 hours, and there is removal of surface glycoproteins from the plasma membrane covering sperm head. And what is acrosomal reaction? Due to multiple contacts of outer membrane of the acrosome and plasma membrane of the sperm head, there is release of proteolytic enzymes which will soften the barrier. And secondly, in the acrosomal reaction, the globular filament, the globular actin, it will undergo polymerization, and it forms a fine needle like filament which will help in piercing the membranes covering the oocyte. So far we have seen how the sperm is transported to the uterine tube in the ampullary cavity and how it undergoes capacitation and acrosomal reaction. Now simultaneously the oocyte is shed from the ovary during the process of ovulation. Okay, so the ovary will shed the oocyte during the process of ovulation, and this oocyte should get fertilized within 24 to 48 hours after ovulation. If it is not fertilized, then it will undergo degeneration. Now, how the oocyte shed from the ovary reaches the ampulla. So, the factors responsible for transport of the oocyte are sweeping action of fibria. Now, we know the parts of uterine tube. There is uterine ostium, then intramural part, then there is ampullary part, infundibulum, and lastly, we have the finger-like projections called fimbria. So these finger-like projections or the fimbria, by its sweeping action, will propel the oocyte into the uterine tube. Again, you know that the uterine tube is lined by ciliated columnar epithelium. So here you can see the ciliated columnar epithelium. So these cilia will again propels the oocyte towards the ampulla. Then there is successive rhythmic contraction of musculature of uterine tube. Also the fluid current and the chemoattractants released by both the gametes will help in the migration of oocyte to the ampulla. And lastly, transcoelomic migration. What does it mean? If the oocyte is shed from right ovary, it will reach the left uterine tube by transcoelomic migration. So these are all factors which are responsible for transportation of oocyte from the ovum, from the ovary to the ampulla. And it should be again remembered, the point to be noted here is the lifespan of ova is only 24 to 48 hours. So it should get fertilized within 48 hours of ovulation, otherwise it will get degenerated. In this first slide, you can see that the spermatozoa and the ova both have reached the site of fertilization, that is ampulla. But before entering into the ova to fuse with the oocyte, the sperm has to penetrate the three barriers covering the ova. So which are these three barriers? The outermost covering or outermost barrier is corona radiata. It is derived from the follicular cells of the cumulus oophoricus. Then the second barrier is zona pellucida. It is an amorphous glycoprotein which is derived from the follicular cells, and it is having striated appearance. The third barrier is the vitelline membrane which is covering the oocyte. Okay, so from outside inwards, the three barriers or the three coverings of the oocyte are corona radiata, then the second is zona pellucida, and third is the vitelline membrane. So all the spermatozoa, means about 300 to 500 spermatozoa, they are reaching the ampullary cavity and all are involved in disintegration of this corona radiata. So how the corona radiata is disintegrated? It is by liberation of hyaluronidase from acrosomal cap. So hyaluronidase is a proteolytic enzyme released from acrosomal cap, which will help in disintegration of corona radiata, so that the sperm will enter into the zona pellucida part. The second barrier which the sperm comes across is zona pellucida. So the zona pellucida is an amorphous glycoprotein which has striated appearance, and it is derived from the oocyte. So this zona pellucida is made up of three glycoproteins, ZP1, ZP2 and ZP3. Now the sperm head, which has disintegrated corona radiata and reached the zona pellucida, will bind with specific receptors for ZP2 and ZP3 glycoprotein. This will induce, now here you can see the spermatic head, the head of the spermatozoa, it is binding with the ZP2 and ZP3 receptors. So this will induce acrosomal reaction, and it will liberate a proteolytic enzyme called acrosin. So this acrosin will digest the zona pellucida, which is present around the sperm head, and it will allow the entry of the sperm into the perivitelline space. So we will revise it again. Here the sperm head has reached the zona pellucida, then it will bind with the specific ZP2 and ZP3 receptors, and due to acrosomal reaction a proteolytic enzyme called acrosin is released, which will digest the zona pellucida. Now what is the function of zona pellucida? Apart from providing protection and allowing diffusion of uterine secretion for the nutrition of developing oocyte, it will also evoke acrosomal reaction. It will allow the interaction between oocyte and spermatozoa. But the most important function of zona pellucida is it prevents polyspermia and it prevents the ectopic pregnancy. So how it prevents polyspermia? It will allow the entry of only one sperm head to bind with it. Once a single sperm head binds with it and enters into the perivitelline space, then the ZP2 and ZP3 receptors over which the sperm head has bind, it will get hydrolyzed so that further no other sperm can bind with it. So it will allow entry of only one sperm into the ova, and thus it prevents polyspermia. Now the second important function of the zona pellucida is it prevents ectopic pregnancy. So now we can see after fertilization a zygote is formed. This zygote will undergo cleavage division up to blastocyst. Up to the stage of blastocyst, the developing embryo is covered by zona pellucida, and it will prevent the binding of this developing embryo at abnormal site in the uterus. So once the blastocyst has reached the upper segment of the uterine cavity where it has to get implanted, at that time on fifth day after fertilization the zona pellucida will disappear from the developing embryo, and it will allow the implantation of this blastocyst over the upper segment of uterine cavity. Okay, so it prevents ectopic pregnancy. As soon as the sperm head reaches the vitelline membrane and fuses with the vitelline membrane, two events occur. First is the rupture of cortical granules, and second is the calcium wave sets in. So you can see in the oocyte towards the periphery, we are having these cortical granules. So these are the cortical granules present at the periphery or the marginal part of the oocyte. So as soon as the sperm head fuses with the vitelline membrane, these cortical granules will rupture, and once they rupture, they will cause hardening of the vitelline membrane and the zona pellucida. Because of this, the other sperms are not allowed to enter into the oocyte. So again, this cortical granules or the cortical reaction will prevent polyspermia. The second event occurring is the calcium wave is set inside the ooplasm. Because of that, the second meiotic division is completed. The secondary oocyte will complete second meiotic division, and it will form a mature ova, and the second polar body is released into the perivitelline space. So in the mature ovum, now we can see the female pronucleus and the male pronucleus. So both these pronucleus will fuse, there is exchange of hereditary material, and finally it will form the zygote. After the approximation and fusion of male and female gametes, fertilization occurs. And soon after fertilization, there is completion of second meiotic division. As we have seen earlier, when the sperm fuses with the vitelline membrane, calcium wave sets in the ooplasm of the mature of the secondary oocyte, and there is completion of second meiotic division. So the secondary oocyte will divide meiotically. So second polar body is released in the perivitelline space and a mature ovum is formed. Second is restoration of diploid number of chromosome. So the male and the female pronucleus both are having haploid number of chromosomes. Soon after fusion, they restore the diploid number of chromosomes. Then determination of chromosomal sex. In case of male pronucleus, we have 23 pairs of chromosome, 22 autosomes, and one pair of sex chromosome, that is X and Y chromosomes are present in male. Whereas in female, again 23 pairs of chromosomes are there, out of which 22 are autosomes, and the pair of sex chromosome has two X chromosomes in it. So if the X chromosome of male fuses with the X chromosome of female, it leads to birth of a female child. And if Y chromosome of the male fuses with the X chromosome of female, it results in birth of a male child. Thus, the sex is determined by paternal side or the male partner. Then initiation of cleavage division of zygote. Zygote is very large cell. So this zygote is soon segmented into small normal sized cells by cleavage division. Then determination of polarity of the embryo. The site from which the spermatozoa enters into the ovum determines the polarity. Then the axis through which it passes maintains the bilateral symmetry of the embryo. So point, an interesting point is that the from the male side we have only the hereditary material reaching the child, but in case of female there is a transmission of hereditary material as well as the cytoplasm or ooplasm is from maternal side, including the cell organelles present in the ooplasm. Also the mitochondria present in the ooplasm is of maternal origin. So once it was asked why spermatogenesis takes place from puberty and oogenesis starts before birth. So it was written that the from the female side we have the entire ooplasm as well as hereditary material. So there is a large contribution from maternal side. So it is a get ready period for a female from means before birth itself. It starts getting ready for the fertilization purpose, whereas in females, sorry, in case of male, we have only the genetic material which is passed on to the offspring. Then second interesting thing, as discussed earlier, sex is determined entirely by the male partner, because in case of males we have one X chromosome and one Y chromosome, and in female both the sex chromosomes are X chromosomes. So there are some methods of assisted reproductive technology. For example, in vitro fertilization. It involves fertilization of ovum outside the female body. Then gamete intrafallopian transfer is there. It involves direct transfer of sperm and ova in the fallopian tube. Then in case of zygote intrafallopian transfer, it involves direct transfer of zygote in the fallopian tube, and intracytoplasmic sperm injection. Here the sperm is directly injected into the cytoplasm. Now we will see the in vitro fertilization. So when a female partner has normal ovarian function, the ovarian cycle is normal, but she is unable to reproduce because there is bilateral obstruction of fallopian tube. In such cases, in vitro fertilization is done. Okay, so a female with normal ovarian function but unable to reproduce because there is bilateral obstruction of fallopian tube due to any cause. There can be stenosis, atresia, infection, or obstruction due to any other reason. In such cases, in vitro fertilization is done. So in this, the the female is pre-treated with the hormones or medicines which will stimulate the maturation of many Graafian follicles in single ovarian cycle. For example, clomiphene citrate, gonadotrophin, then gonadotrophin stimulating hormones. This will all cause uh, the they will stimulate maturation of many Graafian follicles in a single ovarian cycle. After that, the ovarian cycle is studied, and the ova are then aspirated through a cervical, uh, uh, a needle is passed through the cervical canal, and these oocytes are aspirated. Okay, after that, the oocyte is kept in a sterile container having cultural media, and the sperms from the male partner are allowed to fertilize the uh, ova or oocyte in the same cultural media under standardized temperature and pH. After that, the cleavage division will occur, which is assessed under stereoscopic microscope, and when the cleavage division reaches the stage of 8 cell morula, about 3 to 4 such morula, or we can say developing embryos, are then transferred to the uterine cavity. It is known as embryo transfer. So this is possible only for females who have normal uterine cavity, but there is blockage of fallopian tube. Now suppose a female has agenesis of the uterus, or there is adhesion, there is atresia or stenosis in the uterine cavity also. Then in such case, the embryo is transferred to some other female's cavity. So it is known as surrogate mother. Normally, during each ejaculation, about 200 to 300 million sperms are emitted per ml. So more than 15 million sperms per ml is considered normal per ejaculation. But when the quantity reduces from 10 to 20 million sperm per ml, it is called mild oligospermia. Oligo means less, so the amount of sperms will reduce. It is called oligospermia. In moderate oligospermia, 5 to 10 million sperms are there per ml. In severe oligospermia, only 1 to 5 million sperms per ml are present. And the cryptozoospermia means zero to rare sperm, and azoospermia means there is no sperm. The male is completely infertile. Okay, so there is infertility in males known as azoospermia or absence of sperms. Inability to produce a biological child is known as sterility, and inability to conceive after a certain period of time is known as infertility. We have seen these terms gift, zift, and in vitro fertilization and surrogate mother. Now we'll see what is superfecundation. So in superfecundation, if two oocytes from the same menstrual cycle are fertilized by two sperms in the same menstrual cycle, it is known as superfecundation. Means two oocytes are fertilized by two sperms in the same menstrual cycle. That means the age of the fetus, both the fetus will be the same. Okay, so if the male fertilizing it is the same, male partner is the same, it is called homo superfecundation. And if two different male partners are there, then it is called hetero superfecundation. Now, what is superfetation? Here, the two oocytes are fertilized by two sperms, but these oocytes are coming from two different menstrual cycles. So the age of the two fetus will be different. Then the next term is multiple birth. Nowadays the incidence of multiple births is increasing because in case of infertile females, these infertile females are pre-treated with hormones which induces maturation of multiple Graafian follicles in the same ovarian cycle. So as multiple follicles are shed, there is a chance of multiple births. Then the second reason is in case of in vitro fertilization, the fertilization of ova and sperm occurs external to the body. Yes, it occurs externally and it is allowed to fertilize, then cleavage division reaches up to 8 cell stage of embryo or morula. So such embryos, at least 3 to 4 such embryos are transferred to the uterine cavity with the hope that at least single ovum, the single fertilized ovum will get implanted normally. But fortunately or unfortunately, if all the four fertilized ova get implanted normally in the uterine cavity, it will lead to multiple births. Then what is parthenogenesis? Parthenogenesis means when the sperm reaches up to the vital lines membrane, there is excitation of the ova, and the ova without contribution of sperm will start segmenting. It will start cleavage division. And in case of fishes, such kind of reproduction is seen in which there is no contribution from the male partner and the ova itself will divide and it will reproduce. But in case of human beings, there is no viable fetus developed by parthenogenesis. It may lead to ovarian teratoma.